All right, class. We are going to finish off the notes on the neural tissue today. We are in chapter 12, and we just got finished going through the cell types. Okay, and we need to finish the notes starting here. We're going to talk about the way that neurons send a signal from one end of the neuron, the dendrites and the soma, down to the opposite end. Okay, that's the first task. Okay, talk about potentials: resting potential, graded potentials, action potentials. Once we have reached the end of one neuron, then we have to talk about synaptic transmission. What is the behavior, what's the activities of synapses, the neurotransmitters, and the receptors that are involved in two neurons communicating with each other, or one neuron communicating with an effector or a gland. All right. So these are the topics here that we're going to go over: resting potential, graded and action potentials, and then synaptic activity and information processing. We'll talk a little bit about IPSP and EPSPs at the end. Okay. So we've spent a lot of time looking at the plasma membrane of neurons. We said that there's a separation of charge that can occur across the membrane. There are various proteins that exist here, pumps and channels that allow for the movement of individual ions like sodium and potassium. Okay? The overall separation of this charge creates a potential or a voltage. That is, the cell has an overall negative charge, okay, due to around minus 70 millivolts. Okay? And the movement of charge, a wave of depolarization across the membrane, is what a graded or an action potential is. Okay? So a couple properties here, 1 through 3. The extracellular fluid, okay, the ECF here in the gray, and the intracellular fluid vary, okay, greatly in their ionic composition. Okay? So there's more sodium chloride on the outside and more potassium on the inside. Okay? Number 2, cells have selectively permeable membranes. When something is permeable, that means that ions or molecules are allowed to pass through. Okay, and we say it's selectively permeable because only certain ions are allowed to move. And the proteins that exist here in the membrane are very ion specific. Sodium channels move sodium, potassium channels move potassium, and the various pumps with the with the names on them can move only those ions. Number 3, the membrane permeability varies by ion, that is, the channels are specific. Okay, that's number 3. All right, we've said that the neuron is like a banana in the ocean, rich in potassium intracellularly, and the sodium chloride ions are in high concentration outside the cell. Okay, the overall result inside the cell is that it is negatively charged. Okay, so grumpy face for a negative charge. The reason for that distribution is this particular protein. Okay, it is the sodium-potassium ion exchange pump. Okay, it has several names. Okay, sometimes it includes the ATPase there in the name, the sodium-potassium ATPase, or the sodium-potassium ion exchange pump, or just the sodium-potassium pump. Okay, so there's a couple names that you will hear it referred to, but it is one of the most important proteins in the human body. It's right up there with like collagen and hemoglobin and many of the other proteins I say are the most important proteins in the body. But it's because of this protein that all cells maintain this separation of these ions. Okay, sodium and chloride outside and potassium on the inside. This is the clever mnemonic that helps us remember this protein's role. Okay, it's called 3-2-1 Nokia. Okay, Nokia is an old type of phone. The 3, okay, if you line up the 3-2-1 actually underneath the N, K, and A. Okay, 3-2-1. Okay, 3 sodiums move out, okay, of the cell. That is efflux. Okay, 2 potassiums move into the cell, so potassium influx. And both of those activities are directed by the hydrolysis of 1 ATP. Okay, so the A there is for 1 ATP. So it's a very clever mnemonic to remember this particular pump. Okay, 3-2-1 Nokia is for this pump and establishing resting potential. Okay, there's a funny meme here. during action potentials, sodium rushes into the cell. So here's a lady, knocks on the door, hey, mind if I come in? And the guy answers, not at all, do you mind if I leave? It's kind of funny, it's humorous, because sodium and potassium turn out to change places on the opposite sides of the membrane. Okay, we've drawn this picture, okay, we've gotten to know the several proteins that are involved in the plasma membrane. Okay, we talked about the sodium-potassium ion exchange pump. We talked about leak channels. There are potassium leak channels. That is, they're always open a little bit, and they allow for some slow leak of potassium outside of the cell. Okay, those are not drawn here, but they were in the drawing assignment. We also have sodium channels and potassium channels. Okay, we also have chloride and calcium, but they are not shown here. The overall difference across the membrane is negative, and it is approximately minus 70 millivolts. Okay? The negative charge comes from proteins, and they also come from phosphate ions. Okay? Here's another illustration of that. Okay? Here's the charge diagram, or the charge graph, and this is really the sequence of events that occur when we have a graded potential or an action potential, okay? This is really more about action potentials, but it would apply to both, okay? We start at resting, which is at minus 70 millivolts, okay? So green. Then we have some depolarizing event, that is, we go from 70 up to minus 55, okay? The textbooks might say minus 60, but minus 55 or minus 60, that is usually a dotted line right about here. That is called the threshold potential. It is threshold for sodium channels, okay? They are voltage-gated sodium ion channels at 55. If we can get up to minus 55, well, then we can trigger the voltage-gated sodium channels. And then we have a rapid depolarization. That is, we start to go from negative towards zero, so depolarize. And then we actually go past that, we go into the positive territory. Right around plus 30 or plus 40 in this area at the top of the curve is where the sodium channels close, and secondly, the potassium channels open, okay? So the voltage gated here for the sodium channel is minus 55 and plus 30 or plus 40 approximately. And the potassium channels are gated at plus 40 and minus 70. Okay, so the potassium channels are open, allows for rapid repolarization. Okay, we're going to repolarize that membrane. Okay, go back towards minus 70. But these potassium channels are slow to close, so it always overshoots a little bit. We have this trough area of what we call hyperpolarization. So repolarization and hyperpolarization are both in the same direction. Okay, that is getting more negative. The difference is one is returning to minus 70, one is actually bypassing minus 70. Eventually it's going to return up to resting potential, which again is minus 70. You should know each step of this graph and which channel is responsible for each of these changes in direction. All right, you need to memorize this, okay? So this is the membrane here, okay, this is inside the cell, lots of potassium inside, okay, and this A here is for anion, it means negative charge. In this case it'd be the proteins or the phosphates. We'll talk about that in the next slide. On the outside of the cell we have lots of sodium and chloride, okay, represented by these kind of wedge shapes. You'll see this in the literature and other textbooks and other chapters that indicate the width of the wedge indicates higher concentration on this side of the membrane for sodium and chloride, and on the opposite side of the membrane for potassium. Okay, but it's a little bit more complicated than that. Okay, this is a really important graph. Okay, and displays certain electrolytes. Electrolyte is usually an ion, okay, or a small molecule that has a charge. Okay. This line that runs through the middle represents the plasma membrane. Up here would be outside the cell, and down here would be inside the cell. And the box represents the amount of that ion and its relative position. Is it mostly inside the cell, or is it mostly outside the cell? On the left-hand side are cations, that is, they have a positive charge. And on the right side, we have anions, meaning they have a negative charge. So as we've already mentioned, we say that neurons are like bananas in the ocean. Okay, so this yellow bar here represents potassium. It's because bananas have lots of potassium, right? So most of this bar here is represents a lot of potassium inside the cell. Now there's this little bit here that is outside the cell, but it's not much. Okay. On the outside of the cell here, we have lots of the red and the blue. Okay, the sodium and chloride. Now again, there's a little bit of sodium on the inside and a little bit of chloride on the inside, but not much. Okay. Magnesium and calcium are indicated here. They're always a +2. There's slightly more magnesium on the inside and slightly more calcium on the outside. But it's negligible, so we don't really talk about those a whole lot. Over in the anion side, we've already talked about chloride. Okay, proteins and phosphate are these two members here of the anions that have a negative charge inside the cell. They don't really move a whole lot. They're not moving across the membrane like some of these other electrolytes are. Bicarbonate is here. Again, we're not going to talk about it. This relative distribution of ions across a membrane is true in the neuron as well as many other places in the body. Okay, in the kidneys and in the digestive tract. We're going to be referring back to this general distribution of ions all across human cells. So in this graph, there's 4 panels here, and we're moving from left to right in time. Okay, and we're showing in this first panel that resting potential. Lots of sodium outside, okay, and lots of potassium inside. Okay, we have some depolarizing event here where we are triggering sodium channels to open, causing sodium to flood inwards. Okay, that influx of positive charge into the cell is going to cause the cell to go from negative to positive. Okay, so we turn from green to red because we're going in a positive direction. That graph is going to go up. Okay, after which we have this change in potential. The potassium channels open. Okay, and now the potassium is allowed to exit the cell. Okay, so we have a return to negative because we're losing positive charge. And at the end of this, we have sodium and potassium are really intermixed here. Okay, so we've completed the action potential, and now we just have to clean up. Okay, we're going to have this sodium-potassium ion exchange pump just doing its job and returning those ions back into their resting state. Okay, we're going to go from here all the way back to the start, and we're ready to do another action potential. Alrighty. We measure these potentials using a very special tiny probe that we actually insert into the membrane. And historically, these experiments were done in squid great axons. Okay? Squid have this especially large neuron or axon that runs the length of their body. And if you dissect them, a vivisection, you can actually stick a probe into their axon. It's so large that you can actually stick a probe into it. And so this value of -70 or -55 are measured experimentally, okay, using some animals. And it's inferred that humans and all mammals and other animals have similar potentials. Okay? And we've also done those measurements as well, but initially it was in these squid axons. All right. There are some physics concepts here about current and resistance that apply to neurons, but I'm not going to get into it. You can talk about that when you talk about like biophysics. Okay, but we have to talk about the properties that dictate the movement of these ions. Okay, we talk about the electrochemical gradient. Okay, there's 2 parts to that: the electrical part and the chemical part. Okay, the chemical gradient is usually a concentration gradient, that is how much of that thing is inside or outside. The electrical gradient is the relationship of different charges, because opposite charges are attractive, okay? Plus and minus like each other, 2 pluses repel each other. Okay, so both of these forces, they're different forces, but we can apply them both to ions. So potassium is said to have an equilibrium potential. That is, if we allow these ions just to move freely across the membrane, as they will, this is the membrane potential that potassium will achieve, and sodium will achieve plus 66. Why is that? Okay? Why are these the equilibrium potentials for these ions? Let's look closely at that. Okay? First, let's start with sodium. Okay? Potassium. Okay? Starting with potassium. The potassium is orange in this diagram. So here's the plasma membrane. Okay? And we remember that inside a neuron we have lots of potassium inside the cell. Okay? You notice this orange arrow is pointing outward, okay? Because we said that the predominant source of negative charge is these proteins, okay? This orange arrow represents a chemical gradient, or it's the concentration gradient, okay? Notice that there's lots of potassium, lots of orange balls inside the cell, and not many outside the cell. So this arrow represents the diffusion that would occur going from high concentration to low concentration, and therefore it's outward rectifying or outward directed. The white arrow is the electrical gradient. Okay? Again, because this is a positive ion, it's naturally attracted to those negative charged proteins. So it has an attractive force drawing these ions that are outside, actually drawing them inside because of an electrical force. But we have these opposing forces, the concentration gradient and the electrical gradient. So you take the orange arrow plus the white arrow and you add them, you get this small light orange arrow, okay? The addition of these two results in a slight outward force because this outward force of the chemical or the concentration gradient is slightly bigger than the electrical gradient. Okay? These are two opposing arrows and we result in one small force outward. Okay? So this is the electrochemical gradient. Potassium is going to be want to flow outward. Okay? And it is a small effect. It's smaller than sodium. Okay? If we were just to allow them to move freely without, let's say we open up all the channels, all of this would, all the potassium here would flow outward. Because we're removing positive charge, the membrane, the cell is going to get more negative. Okay, so that's how we achieve minus 90. We go from minus 70 to minus 90. Now let's talk about sodium. Okay, sodium is here. We notice that there's lots of purple balls here on the outside of the cell. Okay, because that's where sodium is in high concentration at a resting potential. Okay, we're at minus 70. The inward directed dark purple arrow here is the chemical gradient, okay? Because they're going from high concentration to low concentration, the chemical gradient is going to be inward, okay? The white arrow is the electrical force, okay? Because that positive charge is again attracted to the negative proteins. So that's an attractive force. Again, the arrow is pointed inward, okay? So the dark purple plus the white arrow gets this big light purple arrow, okay? Because they're both going in the same direction. The overall result is a very strong electrochemical gradient, okay? So sodium has more force acting on it than potassium does in the opposite direction. If we were just allow these ions to change directions, we would have a plus 66, okay? Because we would be adding all this positive charge to the cell. We would no longer be minus 70, we'd actually be plus 66. Okay. All right. The reason why we have them separated is because of this guy, that sodium-potassium ion exchange pump with ATPase. Okay, 321 Nokia. All right. So at resting potential, the membrane is highly permeable to potassium, not only because we move those ions due to the ion exchange pump, but also because of the leak channels. Okay, potassium has leaky channels. They're always open a little bit all the time, and they don't change. Okay, they're their own set of channels. There are other gated channels where we have voltage-gated potassium channels, and those are closed or open. Okay, they're not called leak channels; they're gated channels. Okay, leak channels and gated channels are different. Okay, but potassium is more permeable than sodium because of those leak channels. All right. The electrochemical gradient for sodium is very large, but the membrane permeability is very low because there's no leak channels. All right. Okay, leak channels, gated channels. always open a little bit and closed or open. Now, we've already covered this a little bit in class, but let's revisit these three ways of triggering or gating these ion channels. Okay, the chemically or ligand gated, the voltage gated, and the mechanically gated. We have some pictures here. Okay, first the chemically or ligand gated channels. Okay, you can see that there's a binding pocket here for some molecule, okay, some ligand. Well, in fact, this would be some type of neurotransmitter like acetylcholine. Acetylcholine was what we saw at the neuromuscular junction, right? So the neuron releases acetylcholine, it runs past the synaptic cleft and binds to this receptor. That receptor happens to be connected to a channel, okay? Because this is a ligand-gated sodium channel. And the result of that binding is for the gate to open, causing sodium to rush in. Okay, so that's the first type of channel, a ligand-gated or a chemically gated. The next one is a voltage-gated. That is, the reason why it opens or closes is not something binding, but it is a change in the potential. Somehow this protein has a special design which senses a difference in charge across the membrane. And different ion channels are gated at different voltages. Sometimes they're at minus 55, sometimes they're at plus 30, sometimes they're at minus 60. They have different voltages in which they open or close. For sodium, it is gated at minus 55. Okay, it will open at minus 55. The third is, actually before we go to the third, we're going to talk about these voltage-gated channels have kind of three stages, okay, three states. They have closed, but capable of being open. We have open, just regular open, open is open, okay, and we have closed but not capable of opening, meaning it's inactivated, okay? So there's closed and then there's closed asterisk, if you want to put kind of some terminology there. And we cycle through, okay? So once we hit 3 will eventually turn back into 1, okay? So we start here at rest, okay? We have a change in voltage and it will open, and then we have a closed gate, okay, a closed signal, which will get us closed and it's inactivated. And eventually that inactivation goes away and we just go back to regular closed. And we can start the procedure again. Okay, so here we are, the gate is closed, we're at resting potential. Okay, sodium cannot pass through. Okay, let's say the membrane potential changes and suddenly we're no longer at minus 70, but we're at minus 55. Okay, suddenly this gate opens because it's sensed a change in the potential. Okay, so the sodium ions are allowed to flood inward. Okay, as a result of the change in potential, because we're adding positive charge, okay, it can become inactivated. Okay, it actually looks like this rather than the two gates that I showed previously. It's actually a little bit more complicated in how the protein works. They have this kind of ball and chain mechanism, which is a little bit unique. And then the protein will cycle back to this starting stage where the ball will drop out and the gate will reclose. Okay, so these are the three stages that are listed here. All right, mechanically gated is the third type. We talked about ligand gated, we talked about voltage gated, and now the third type is mechanically gated. And this is simply initiated by some physical pressure, okay, a mechanical pressing or stretching of the membrane. You think, well, where does that occur? That occurs in like touch receptors. Meissner's corpuscles and Pacinian corpuscles have physical pressure on them, which allows for a slight opening of these channels, and therefore that's the reason why those pressure touch receptors get activated, okay, mechanically gated. Okay, so here they are. Here's a Merkel cell, here's a Meissner's corpuscle or a Pacinian corpuscle. These membranes have mechanically gated ion channels in them. And so if you press on them, they will cause depolarization, okay? And the axon that is touching them will get activated and it'll send an action potential. There's lots of different receptors, okay? Some of them here, like the Merkel cell or the Meissner's corpuscle, are that mechanically gated, but not all of them, of course. Okay, you have a photoreceptor. Here's a rod from the retina. It's going to receive light stimulus, okay, wavelengths of light. Okay, olfactory is going to be a chemosensation. Okay, taste receptors and olfactory receptors are chemicals binding to them. Okay, hair receptors are again mechanical receptors, but they're a different type. So we'll come back to these other types when we talk about special sense receptors. Okay, but the mechanically gated apply to like the Merkel, the Meissners, and the corpuscle. All right, so we've been looking at the various channels and proteins which establish resting potential, and we're getting to this idea of action and graded potentials, how we get from one side of the neuron down to the other side. Okay. So first we're going to talk about graded potentials. Okay. Now, graded potentials exist, whoops, let's go back. Graded potentials exist here in the dendrites and across the soma, or the cell body. Okay? Only from the tips of these dendrites down to the soma, about to here, that's where graded potentials occur. From the axon hillock, this region here, down to the end, that is where action potentials occur. Okay? If there's no action or graded potentials, everything is at rest, resting potential. The distribution of these important channels, the voltage-gated versus the ligand-gated, is the reason why we have graded here and action potentials here. So let's start talking about graded potentials. Okay? They're also referred to as a local potential. And what I want you to think about for graded potentials is this analogy. Okay? Dropping a small rock into a calm body of water. Okay, when you drop the rock, it creates these ripples, okay, that go out into all directions. And as those ripples go off into all directions, they diminish in size. They get smaller, okay, they get weaker. That's what graded potentials are like, okay? They go in all directions, okay? They diminish in size over distance, okay? The further they go, the weaker they get, okay? And they're the strongest in the middle, okay? If you have a big rock, you'll get a big ripple. If you have a little rock, you'll have a little ripple. Okay, so they're graded. The effect changes with the stimulus. Okay. Graded potentials occur because of sodium depolarization. Okay, sodium channels opening are the result of graded potentials. Cause graded potentials, I should say. Right. So here we zoom in on this little section here of the cell body. It could be in the dendrites, but here we've chosen to highlight the cell body. And at rest, okay, we can measure -70. That's resting potential. Okay, lots of sodium outside, and the other ions are not shown. Okay, so we initiate a wave, a depolarizing event. Okay? We do depolarize, we move the graph towards 0 millivolts, from -70 up to 0. Okay? The movement of sodium through the channels causes that. And so we have this ripple effect of sodium depolarization that's spreading in all directions. Okay? So here we have a rushing in of sodium ions. Okay? Typically, this is where a synapse is occurring. Okay? So a synapse is right here. There's another neuron touching this cell body and is causing a depolarization event right here. Okay, now my finger touching it right there would cause this ripple of depolarization. Okay, if it's excitatory. So here we have the ions flooding in and then running down the membrane. So we have a graded potential initiated here, and it's spreading out. Okay. So there's kind of 4 or 5 key points here that you need to know. Number 1, the effect decreases with distance. As I just said, okay, the further away from the touch point you go, the smaller the waves are. Okay, so it degrades with distance, it gets smaller and smaller. Number 2, the effect spreads passively due to local currents, meaning it will, it's the reason why it kind of peters out, okay? because it's a passive. It just means that it is not propagated continuously. Okay. Number 3, the graded change in membrane potential could be positive or negative, meaning it could be excitatory or inhibitory. Okay. Depolarization means that the direction of the charge graph is to move up. So you can see this red bump at the top here. We are moving closer to the threshold, which would be up here. Okay, so that would be a depolarizing event. The hyperpolarizing means we're getting more negative, so we're hyperpolarizing. Okay, so that's this would be excitatory, meaning we're moving closer to the threshold potential. Here, this would be inhibitory. We're moving further away from the threshold, which would be up here. Okay, so it can be positive or negative, excitatory or inhibitory. That's number 3. Number 4, okay? The stronger the stimulus, the greater the change will be. Okay, so at the source, if we increase the strength of the depolarizing event, we get more depolarizing, we can move that wave further across the soma or the dendrites. Okay, stronger goes further. That's important. Okay, so in this graph, you understand that this is the resting potential. Any movement of the line upwards we would call depolarizing. Okay, any movement of the line downwards we would call hyperpolarizing. Okay. This is excitatory. Okay, this is inhibitory. That's important. So here's the terms that we will come back to in just a little bit: an EPSP or an IPSP. Okay, now PSP stands for postsynaptic potential. Okay, and it's always the same. So really, the only letter here that means anything is the first letter. Okay, E is excitatory, and I is inhibitory. Notice for the excitatory, the bump is going up, so we're getting closer to threshold, which is the green dotted line. Whoops, I don't want that. And over here, for the inhibitory, well, the bump is going down, so we're actually going further away from threshold. Okay, so it's inhibitory. So what causes excitatory postsynaptic potentials? Well, sodium influx. Okay, sodium is the one that causes depolarization. Okay, so sodium channels opening will always cause an excitatory postsynaptic potential. What causes this to go further away? Well, potassium channels opening, because remember, potassium's on the inside. If we're losing positive charge, that means we're going to get more negative. Chloride also has the same effect because we have more chloride outside the cell than inside. So that chloride rushing in will again increase the negative effect. So we're going to have an inhibitory or a hyperpolarizing effect for potassium rushing out or chloride rushing in. That's important. All right, so here's a picture of a graded potential. Here's neuron number 1 synapsing here with neuron number 2. Okay, and we're on the cell body here. Here's a dendrite, here's a dendrite, here's a dendrite. We have to head towards the axon hillock, which is off screen. Okay, so right here we have the strongest depolarizing event because the neurotransmitter here is opening sodium channels. Okay, and the red color here indicates the strength of depolarization. And as we move further and further away, well, we get this decreasing effect of that depolarization. So it's less and less strong depolarization. And it's going in all directions. Okay, so here's the graph, here's the center point. If we go in any direction, we can see there's a decrease in strength. All right, so again, those different points here, graded potentials, they're omnidirectional, they go in all directions. Okay, the bigger the rock, the bigger the wave, meaning you can have a graded, okay, that is bigger produces bigger, smaller produces smaller. Okay, they can be plus or minus. Okay, they can be excitatory or inhibitory. Okay, green means go, red means stop. Okay, so or inhibit. Distance is important. Okay, so how far you are away from the synapse to the axon hillock. If that's a short distance, okay, that's meaningful. If it's a long distance, if you have a really long dendrite to the axon hillock, well then that's unlikely to get the same response, okay, because it has to travel a longer distance. Okay? And the location here is in the dendrites and soma, which we've already talked about. All right, and so here's the sequence of events: depolarize, repolarize, hyperpolarize, that we can see here with the graph that we've already shown. Now, that's in contrast to action potentials. What we just talked about was a graded potential. Now we're going to talk about an action potential, okay? Action potential, I want you to think of a different thing. Here we're talking about not dropping a rock into a pool, okay, causing ripples. I want you to think about tipping over a set of dominoes, okay? Now, assume that all the dominoes are set up perfectly, they're perfectly lined up, and so if we tip over that first domino, we could be guaranteed that the last domino will tip over with the same force, okay? So it's an all or none process. As soon as we tip over that first one, if we initiate depolarization here, it's gonna go brrrr all the way to the end, okay? It's not gonna grow, it's not gonna diminish like graded potentials. It's gonna stay the same force all the way through, okay? And it also goes in one direction, okay? It doesn't go in many directions, it only goes one way, okay? Down the axon. All right, this is what you think about. The dominoes falling over is action potentials. Okay, here's the graph. We've talked about this already. Okay, depolarization to threshold. Okay, this is all happening at the axon hillock. Okay, the trigger zone is the axon hillock. That's where action potentials are initiated. Okay, this first couple dominoes, okay, would be the axon hillock. All the rest of them would be lined up going down the axon. We first start with sodium channels, and then we open the potassium channels, and then afterwards we return to resting potential. Okay, so here's that graph that we've drawn. Now, layering on top of this are a period of time which is called the refractory period. Okay? The red and the purple here are both refractory periods. One is called an absolute refractory period in red, and in purple is the relative refractory period, okay? The absolute refers to the big wave here, okay? From approximately threshold, minus 60, minus 55, up and then back down to, again, approximately threshold. The big wave up is the absolute refractory. This little trough area here is what we call the relative refractory, okay? So in this area, we cannot initiate another wave of depolarization because it's already initiated one, okay? So it cannot start another one. Whereas when we're here, if we give a big enough stimulus, a big enough wave of depolarization, we could trigger another one. We would have to get from minus 90 here up to threshold. So it's a further distance to go, but if we had a big enough stimulus right here, we could force it to go. Okay, so we call it relative refractory, not absolute refractory. In the red zone, not happening. We can't do a second one. In the purple zone, it's going to be tough, but we could send another one if it was big enough. Okay, so here is a way of illustrating that. Okay, here's an axon. Okay, shown here in these kind of chopped up sections. In green is resting potential. Okay, minus 70. In the red is this wave of depolarization that is traveling down the axon, like the football stadium people doing the wave, right? These people start doing it, and these people start doing it, and these people start doing it, etc. The ions are switching spots, and we have this wave of depolarization. On the trailing edge here is actually the refractory, the period of refractory. In red would be the absolute refractory period, and in yellow would be the relative refractory period. And so the upshot of the refractory period is that we get unidirectional conductance, okay? So let's walk through these points here. Action potential is a wave of depolarization. That's really the definition of action potential. I should add on here wave of depolarization down an axon, okay? Down an axon. Followed by a refractory period, okay? Shown in the red and the yellow. Okay? resulting in exclusively unidirectional conduction. That is, we're moving the signal only in this direction. Okay? Action potentials can never go backwards. Okay? Don't confuse this with anterograde and retrograde transport. That is moving of molecules up and down the axon internally. That has nothing to do with electrical charge. Okay? You're just hauling stuff in the cell. All right, so here are the steps in an action potential. I'm not going to walk through them. Talked about the refractory period, absolute and relative. Okay. Now, when we talk about action potentials, there's two types. Okay, there's continuous propagation and there's saltatory propagation. And this really has to do with our good friend myelin. Remember myelin? Myelin is a result of oligodendrocytes in the CNS and Schwann cells in the PNS. If you're myelinated axon, you do saltatory. If you are unmyelinated, you do continuous. Let's take a look at that. Okay, on the left here would be kind of a time series from top to bottom. We're starting here, and then here's next, and here's next. Okay, we're moving just a short distance. Okay, this is a continuous conduction. Okay, because there's no myelination here. Okay, the exchange of ions has to occur at each place along the axon from the beginning all the way to the end. Okay, that's slow. If we have myelin present, here's a couple Schwann cells, okay? And in between the Schwann cells, we have these little nodes, okay? It's where there's a tiny little gap between the Schwann cells. We call that the node of Ranvier, okay? The node of Ranvier. It's a French word, okay? Named for a French scientist. And what happens in between these nodes is that there's a jump. For some reason, there's a jump in depolarization. And what we think is that the positive and the negative ions are kind of bumping into each other. Okay, and they kind of hit each other and they bump all the way down this myelinated section, and we see this wave of polarization jump from here to here. The upshot of this is that it's faster. Okay, jump, 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 and we get this faster conductance in myelinated axons compared to unmyelinated axons. This is called continuous. This is called saltatory. Saltatory for assault to jump. All right. So here's that in the continuous fashion, moving down the axon. And then the saltatory, if we have myelinated there, jump, jump, jump. Okay, so it's faster. Alrighty. Oops, I'm going backwards, sorry. Hit the wrong arrow button here. Alright, we just talked about myelination, okay? Talked about saltatory conduction. But there's, and so that influences the speed of conduction, okay? How fast can a signal move down the axon? That's quite an important topic. And it turns out that not all axons move at the same speed, okay? There are actually kind of three flavors of axons. You have the really super fast, you have kind of the medium fast, and then you have the slow. And there's differences in their organization. The first difference you'll notice is that the presence or absence of myelin. This yellow sheath here is the myelin, okay? It's just one coating, so you can say this is a Schwann cell or an oligodendrocyte. The second feature that you should notice is the diameter of the axon. Here the diameter is larger, so it's a thicker axon, and this C fiber is a much more narrow diameter, okay? So two factors that influence speed: myelination, okay, and the thickness of the myelination. Here it's heavily myelinated, here it's just minimally myelinated, and here no myelin. We call these the A fibers, the B fibers, and the C fibers. Okay, and actually A fibers have several different subtypes in them. And so we can see that they correlate with each other, okay? You're always gonna have a big diameter with lots of myelin. Those are gonna be the fastest fibers, okay? Narrow diameter with no myelin, that's gonna be the slowest fibers, okay? Slowest axons. All right. So what is the relative speed here that we're talking about? About 140 meters per second. Okay, so think about a football field. A football field is 100 yards, okay, give or take, 120. Okay, so if we can run down a football field in 1 second, okay, That's about how, it's like 1.5 football fields. Again, 1.25, whatever. Okay, but that's pretty darn fast, like, okay, quick. Okay, meters per second. So that's as fast as some of the fastest fibers can go. The A alpha, the A beta, and the A delta are some of those primary afferent neurons. Okay? So those fast fibers, okay, are going to be things like taking in vision. Because when we see things, we need the fastest signal for vision. Because as our retinas are taking in fresh information, it's like zip to the back of the brain as fast as it can go, okay? As fast as the fibers can take it. B fibers are lightly myelinated, okay? And they have a medium-sized diameter, and they go about 18 meters per second. So we've dropped by about a factor of 10, from about 140 to 18. Okay, so think about 100, 10, and 1. Okay, because the C fibers here, the last one, okay, C fibers are only 1 meter per second. So think about 100 meters per second, 10 meters per second, 1 meter per second. About 10-fold change drop in each of those. Okay, so involuntary muscle, gland controls, and some pain receptors. Okay, now this has an interesting application because if you think about applying like lidocaine. Lidocaine is what you'd use to like numb nerves if you're doing like dental work, okay? And it turns out that some pain receptors are C fibers, which just sounds kind of weird. You'd think pain would be a fast fiber. It's not, okay? They're usually the C fibers. The good thing about this is that that lidocaine can penetrate very quickly and efficiently into those C fibers. So it gets numb very quickly. So that's a benefit when you're having surgery, when you're having dental work done. Okay? These larger myelinated and thicker diameter fibers, they're more slow to uptake the pain medication. Okay? So there's pros and cons to the arrangement here clinically. All right, so we've talked about potentials. Okay, we talked about resting potential and graded potentials and action potentials. Now we're going to talk about how do we communicate from one neuron to another neuron. Okay, we're going to talk about the synapse. Okay, what are synapses? How do they work? And what are the types of neurotransmitters and receptors that regulate the activity in synapses? So some terminology, okay? At a synapse, we're going to have, synapse is two structures sending a message, okay? It's usually a neuron with another neuron, or a neuron with an effector, such as a muscle, or a neuron with a gland, okay? And we'll always have a synapse at the end, okay? The synapse will have a axon terminus, okay? This is the axon terminal. Axon terminal contains these vesicles. The vesicles contain neurotransmitters, okay? Neurotransmitters like acetylcholine, okay? It's usually a collection, a little ball or a sphere of neurotransmitters, and if they receive an action potential, they can get stimulated to then migrate and perform exocytosis, releasing their payload into the synaptic cleft and then binding to a cognate receptor. That is the appropriate receptor for the acetylcholine, or the neurotransmitter. So we call this neuron the presynaptic neuron. Presynaptic because it's the first one. It's before the synapse. This neuron down below is called the postsynaptic neuron. That's important because I'm going to use pre and post. Okay? Pre is the first one, post is after the synapse. As I said, we can have multiple types of synapses. Okay? Between two neurons, between a neuron and muscle, or between a neuron and a gland. All of these have synapses and they all behave in roughly the same way. We call these neuromuscular junctions, we call these neuroglandular junctions, okay? And these we call neuron-neuron junctions, or just regular synapses. Now, kind of a special topic, okay? You might be aware of THC and CBD. Okay, CBD, these are cannabinoids. Cannabinoids are a class of molecules that can bind the cannabinoid receptor. Okay, they have two types of cannabinoid receptors, CB1 and CB2. Okay, and this is THC is one of the main ingredients or components of marijuana. Okay, marijuana has these chemicals inside of it. If you burn it, you oxidize it, and you inhale it, then it gets systemic, and this functions like a key that unlocks that receptor. Okay, it doesn't unlock it, but it binds to it and it activates that receptor. These receptors are located in different parts of the body and have various neurological effects. Okay, of course, a plant chemical is not the natural ligand. It's not the thing inside of us for which this receptor was designed. There are native ligands that are inside, which we'll talk about. But it just so happens that this drug, this plant, has this chemical, and if you smoke it or otherwise take it, CBD oil is a different type of ligand. It does not have the same effects as THC, which is only if you like smoke marijuana. Okay? CBD oil, okay, or CBD, is an agonist, but it works through a different mechanism. Okay, so you don't get those mind-altering effects with CBD, but you do get that with THC. The receptors are located in slightly different areas. In the brain is where THC occurs. You get that mind-altering effect. Here's a really complicated diagram showing you the CB1 receptor. It's on a presynaptic membrane, not the postsynaptic, but the presynaptic. Here are these native ligands, okay? 2AG and anandamide, okay? These are native ligands, that is, they originate within humans, okay? Here is THC, okay? That is a foreign ligand, and it comes in from the blood supply after inhalation, and it binds to this receptor, and it creates a change in a certain class of neurons. I'm not going to get into that much detail because frankly it's really, really complicated. It's beyond the scope of this class, but in synapses is where we have the ability to really modulate neurological activity, okay? Like the effect that marijuana can have, amongst many other drugs that are legal and appropriate. All right, we have two general classes of synapses, okay? We have chemical synapses, and those are the ones that we're talking about right now. Then we have electrical, and we're not talking about electrical right now. These occur in the cardiac cells, okay? Cardiomyocytes have electrical, and those are based on gap junctions. Let's take a look at the image here, okay? This is a chemical synapse. Here's our vesicles. They're full of neurotransmitter. They open up, spill into the synaptic cleft, bind to a receptor, okay? It could be an ion channel or it could be a G protein coupled receptor, either way. This is a chemical synapse. Over here is an electrical synapse, okay? So we have an electrical signal that's running down this cell, and it's physically coupled through these gap junctions, okay? Now a gap junction is a set of proteins that creates a kind of a tunnel directly connecting cell number 1 to cell number 2, okay? This tunnel jumps the gap. The thing that jumps the gap here is the neurotransmitter, okay? There's no direct connection here. There's a gap, there's a synaptic cleft. So chemical synapses have different dynamics than electrical synapses. Okay? This is really a direct effect. It's like you go right from the cytoplasm here directly into the cytoplasm of the second neuron, okay? Or in this case, more appropriately, these would be two heart muscle cells, okay? The flow of electrons or calcium ions down the heart tissue. We'll learn about that in the cardiovascular system. All right, all right. Neurotransmitters can have two effects. You can have excitatory effect, or you can have an inhibitory effect. Inhibitory, okay? Excitatory is always causes depolarization, okay? Green means go, so I color it green. Green means go. S means stop. Red means stop, okay? So hyperpolarization is we're going to inhibit the signal, okay? Green, depolarization means we're going to send the signal. So we're going to talk about the role of acetylcholine. The effect of a neurotransmitter on a postsynaptic membrane depends on the receptor. Okay, this is a key point. It depends on the receptor, not on the neurotransmitter. Okay, for example, acetylcholine, we typically associate acetylcholine with an excitatory response because that's what we see in skeletal muscle. It is excitatory. Green means go, so it's promoting an action potential. However, in some cases, acetylcholine can have an inhibitory effect because the receptor it binds in these special junctions in the cardiac tissue has an inhibitory role. So what does that mean? It just means that acetylcholine is not just like bar none, always excitatory. Okay, it's always going to send a signal. That's not what it means. Okay, acetylcholine is mostly excitatory. Green means go. Okay, but sometimes it can have an inhibitory effect. It just depends on the receptor. Okay, not what acetylcholine is. All right. We have two general classes of receptors. We have cholinergic and adrenergic. We're going to talk about these two major classes. There's others. We have dopaminergic and others. But for now, cholinergic and adrenergic are the two that we're going to talk about. Cholinergic gets its root from acetylcholine. So the choline part here is the cholinergic. It just means that it's binding acetylcholine. All our neuromuscular junctions with skeletal muscles have cholinergic receptors. All our neuron-to-neuron synapses in the peripheral nervous system are cholinergic. And all the neuromuscular and glandular junctions in the parasympathetic are also cholinergic. So it's everywhere. Okay? Acetylcholine is a very common neurotransmitter in the peripheral nervous system. Okay? In the peripheral nervous system, it is one of the most common. Now, in the central nervous system, there are other more prominent. We'll talk about things like serotonin and dopamine and glycine and GABA. Those are much more common, or glutamate. Okay? But in the peripheral nervous system, acetylcholine is our guy. All right. Now, we've kind of covered this material already in the muscle chapter when we talked about a motor neuron in the muscular neuromuscular junction, okay, the neuromuscular junction. We have an incoming excitatory action potential that arrives here at the axon terminus, okay? There are voltage-gated calcium channels that open, causing an influx of calcium. That influx of calcium, the increase in calcium concentration, causes these vesicles to then migrate towards the axon terminus, the synaptic cleft, and perform exocytosis, releasing acetylcholine into the cleft and then binding to a ligand-gated sodium ion channel. Okay? So there's a whole sequence of events here that you need to know. Make sure you understand those events. Okay? So neurotransmitter gets released, binds to the receptor. That binding event causes the channel to open. Okay? It goes from a closed conformation to an open conformation. The result of the open channel is sodium floods in and initiates a depolarizing event. Okay? And then we have this depolarizing event. These little red arrows are going off in opposite directions because this is the beginning of a graded potential. Okay? It's like the drop in the water. We have that rippling effect in all directions. This is going this way, and this is going this way. Okay? This is a graded potential. The beginnings of a graded potential. It starts as synapses. Soon after the neurotransmitter has done its job, well then along comes an enzyme, acetylcholinesterase, and cleaves that neurotransmitter and renders it non-functional. Okay? So acetylcholine, once it's cut, okay, it can get reabsorbed into the presynaptic membrane, okay, and we can resynthesize it and form it into new vesicles, and we're ready to go for a second go round, or a third or a fourth. Okay? So this process happens all the time. We use it, we send the signal, and then we recycle it right away. Okay? There are astrocytes, okay, glial cells, that can also suck up these neurotransmitters and pull them out of the synapse. Okay? A really key principle in neurobiology is how long do neurotransmitters have to act in the synaptic cleft. Okay? That's quite important. If it happens for a long time, we get lots of stimulation. If it happens for just a short time, we get just transient stimulation, only a little bit of stimulation. Okay? So if we can lengthen the amount of time that these neurotransmitters are active, we can expand a response or decrease a response. Okay? So certain drugs act at the synapse to shorten or lengthen the activity of neurotransmitters in the cleft. Sometimes by blocking receptors, sometimes by blocking the reuptake receptors. Okay, there's actually a receptor here that brings this molecule back up into the presynaptic membrane. We can block that if we want to. Okay, there's different mechanisms. Okay, synaptic fatigue can occur when there's so much stimulation that acetylcholine can't get recycled fast enough to keep up with the demand. But it rarely occurs. Okay, how many neurotransmitters are there in the body? Lots of them. Okay, over 50 neurotransmitters other than ACH. Okay, these are all the different classes of neurotransmitters. Okay, these are some of the common ones that you're going to see: norepinephrine and epinephrine, otherwise known as noradrenaline and adrenaline. Adrenaline and epinephrine are the same thing; they're synonyms. Dopamine and serotonin; those are some big ones in the brain. And then GABA, gamma-aminobutyric acid is the long name here. Gamma-aminobutyric acid. We just shorten it to GABA. And what's GABA is famous for is it is a very common inhibitory neurotransmitter. Okay? It binds to receptors that are almost always, I want to say they're always inhibitory. Okay? They're binding to chloride and to potassium receptors. All these other ones have a mixed function. They could be plus or minus. So, epinephrine, typically it's excitatory, okay? It is part of the adrenergic synapses, okay, because it's releasing epinephrine, which is the same as adrenaline. Adrenaline and epinephrine are the same thing, okay? So adrenergic is the same as adrenaline. Dopamine can be excitatory or inhibitory, okay? It's important. It's involved in Parkinson's disease and cocaine use, unfortunately. Serotonin, very important for attention and emotional states, okay? It can be plus or minus. Gamma, aminobutyric acid, GABA is always inhibitory, okay? It's always inhibitory. Its function is inside the CNS. Neuromodulators, okay? There's a lot to be said about neuromodulators. I am just going to kind of coast over top of it and not spend a lot of time here. But there are other chemicals released in the synaptic terminals similar to the function of these neurotransmitters, okay? And they can modulate how those neurotransmitters function, okay? It's a class of chemicals that can modulate how neurotransmitters work. That's all I'm going to say. Neuropeptides and opioids, those are all classes of neurotransmitters. Okay? Now, we talked about neurotransmitters. Now let's talk about receptors just a little bit. Okay? There's generally two classes of receptors. Okay? There's a third here that has to do with nitric oxide. We're not going to talk about nitric oxide a bunch because I'm already short on time. Okay? We have some receptor types have a direct effect. Okay? And some receptor types have an indirect effect. Okay, let's dig into that a little bit more. This will be the last topic. Okay. These direct effect receptors are what we call ionotropic. Okay, ionotropic. Okay, so the key word here is ion. Okay, ionotropic. And these types of receptors are gated ion channels. When I show you a picture of them, you'll immediately know what they are because we've already seen it with acetylcholine. Okay, acetylcholine is a ligand-gated sodium channel ligand. The other class is the indirect effect receptors. Okay, they're called metabotropic. Holy moly. Okay, metabotropic. Okay, metabotropic functions through a different type of receptor. It functions through what's called a G protein coupled receptor. Okay, G protein coupled receptor. You might see it abbreviated as GPCR. GPCR is a broad class of receptors that can bind neurotransmitters, amongst other mechanisms. Okay, so ionotropic and metabotropic. All right. I'm going to pause right there. You guys are probably saturated with this information, so I'm going to come back, and when we come back, the next lecture, we will continue talking about these various types of receptors. Okay? See you in class.